Hello. Hi, everybody. Good evening. Who all are in this time frame, and good morning to those who are outside the time frame. I'm really delighted. I'm Dr. Monica Kaushal, consultant neonatal perinatal medicine in Emirates Specialty Hospital, and I'm very delighted today because we are running a fourth symposium under the patronage of NNF IUA branch chapter, and we have been successful in theme kind of a symposium, and today our theme is NEC. I think everybody of us is dreaded when we think about NEC, and the good thing, and we are fortunate to have speakers who are really working on NEC day in and day night, and so they will be able to give us a real scenarios where how we will be able to take care of these tiny babies when they have this kind of a problem. So we will start with Dr. Erica Cloud, who will be talking on the NEC, whether it is treatable or preventable, and then we will be having Professor Rakil, who will be talking on every time when we are in confusion whether we can give transfusion or not. And then we will have question and answer sessions. So I would like to be introducing Dr. Erica Cloud, who is professor in University of Chicago in Department of Pediatrics and Medicine. And as you can see, she is actually heading neonatology and gastro. So you can see her interest, how strongly she has interest on the gastro part, and she's done all her research on the microbiomes and the NEC. And so we would be actually listening from the horse's mouth. And she has lots of publications, and she has a lot of grant because of these publications, and she has leadership roles in the AAP co-chair for the Central Conference on the Perinatal Research term, and she's an executive board in the Family Institute, Burroughs Wellcome Fellowship, Best Prep Executive Committee, and Faculty Leadership Cabinet, Domitius Family Initiative. So actually, if I continue her CV, it's a big 5 to 10 page CV. And so we are really honored to have you, and you are accepted our request to be with us in spite of your very, very busy schedule. Thank you, Dr. Cloud. And we would like to listen to you whether we can treat or we can prevent the NEC. Floor is all yours.
Thank you for that kind introduction. I'm going to go ahead and share my screen. So it's a pleasure to be here today. As mentioned, my interest is the microbiome. And so I will have to tell you from the beginning, I don't have all the answers regarding prevention and treatment, but I would like to present my work on the microbiome and put into that context the topic of probiotics, which I think has come up quite a bit, and just sort of share a little bit about how I'm thinking about these issues. So I'll just jump right in with some background to kind of get us all on the same page. I know these are things that we're all familiar with, but I think as we go through this, you'll see how this context plays in. So as we all know, necrotizing enterocolitis is an inflammatory bowel necrosis that primarily afflicts premature infants after the initiation of enteral feeding. We know that the majority of patients are born preterm. In the US, our incidence is about 10% of babies born with a birth weight less than 1,500 grams. Now, the textbooks say that NEC classically appears between 7 and 14 days of life, but we all know that susceptibility is inversely related to gestational age. So the more immature you are, the more likely you are to have this disease and the longer window in which you're susceptible. So this really suggests that there's a developmental component to this disease, and there is no race or sex predilection. The pathognomonic feature is pneumatosis intestinalis, which is air tracking in the bowel wall, and this represents bacterial fermentation of intraluminal substrates. We don't have specific therapy because we don't yet know specifically what causes this disease. What we do provide is supportive care. So we put the bowel at rest, cessation of enteral feeds, nasogastric suction. We support with fluids and antibiotics and parenteral nutrition. And then there's surgery if there's a specific indication like pneumoperitoneum, peritonitis, or intestinal obstruction. But our outcomes remain poor. Mortality between 30 and 40%. Particularly for those who require surgery, risk of stricture, short gut, poor nutrition, parenteral nutrition-induced cholestasis, and then certainly risk for poor neurodevelopmental outcomes. So for babies who have severe NEC, those who require surgery, there's increased risk of cerebral palsy, a mental developmental index less than 70, physical developmental index less than 70, any neurodevelopmental impairment. Surgical NEC is associated with increased risk of periventricular leukomalacia and decreased growth in all parameters. So you can see from our non-specific therapy and the remaining poor outcomes that there's still a lot we don't know about this disease. What we do know is that there's specific risk factors. So prematurity is the biggest risk factor, along with bacterial colonization, enteral feeding, and hypoxia or altered intestinal blood flow. And I'm going to focus my talk today on prematurity and bacterial colonization. And for a long time in neonatology, I think we've thought about bacteria as the enemy. So everything is in the context of pathogens and how do we identify them and eradicate them. Just to remind all of you, when we're thinking about pathogenesis, if a microbe is thought to really cause a disease, it's supposed to fulfill Koch's postulates, which is that a microbe suspected of causing a certain disease must be found in every case of that disease. The suspected microbe must be isolated and grown in the laboratory outside of the host. When this suspected pathogen is introduced to a healthy susceptible host, that host must develop the disease, and that same pathogen must then again be obtained and re-isolated from that experimental host. So clearly we do not know the pathogen that causes NEC. On the other hand, there's this idea of commensal organisms, beneficial organisms. So Elie Mechnikoff in 1882 discovered phagocytes, but in 1907 was one of the first to really suggest the possibility of colonizing the gut with beneficial flora. And he is credited with the introduction of the concept of probiotics. So now we have this tension between maybe good and evil. You have good microbes, you have bad microbes. There's actually a third way of looking at microbes, and that is how we think about the microbiome. And this is the community of all the microbes in and on the human body. There are about equal numbers of bacterial and human cells. So bacterial cells really form a significant population in terms of genetic content, metabolic contribution, and it really is important to recognize that human health depends on microbiota health. And if this is true, we then really need to understand the microbial community perspective. So the microbiome is an ecosystem, just like any other ecosystem, a forest or a pond. The goal is to maintain equilibrium. And in a healthy ecosystem, there is high diversity of species, there's balance, functional redundancy, and therefore resistance to disease. But in a sick ecosystem, there's low diversity of species, imbalance, functional disability, and susceptibility to disease. So when we think about the microbiome and microbes, I just want to put this up there as an illustration of community dynamics. So let's imagine 3 organisms, A, B, and C. They're not pathogens, they're not commensal. We're not talking about them as good or evil, just 3 organisms. And let's just imagine that A produces something that B needs so that B can grow. And let's imagine that B and C share a substrate. And so by the increase in B, there's increased competition for this substrate. And so there's less of C. And let's assume that this is what balance looks like. Maybe increased numbers of A and B and decreased numbers of C. What happens if we perturb this community? Let's say we introduce antibiotics and we take out A. So now A is no longer producing the substance that B needs, and B cannot grow. B no longer competes for the substrate, and now C is a greater player in this community than previously. So you can imagine how C may not be labeled as a pathogen, but could have now a different effect on the host. Or let's imagine the situation where there's an altered diet. If you remove the substrate that B and C needs, so that these organisms now disappear, the substance that A produces is now present without B to consume it. And this substance could now affect the host. And you can imagine now in this scenario, A may not be a pathogen, but could cause untoward effects on the host. There's a few more of them. So this situation is called dysbiosis, when there's lack of diversity, loss of keystone species, and overgrowth of opportunistic pathogens. And this is a situation that leads to disease. And it was hoped that by thinking about the microbiome in this way, we could better understand the role of the microbiome or bacteria in necrotizing enterocolitis. So this is where I started my work. And I asked the question, can microbiome analysis be used to identify the bacteria associated with NEC? And for this initial study, I looked at 20 patients, 10 with NEC and 10 control patients. And I happened to have 4 sets of twins where one baby had NEC and one did not. We looked at fecal samples from these babies just prior to the onset of NEC. So a sample was taken, we had been collecting longitudinal samples, and we selected the sample just prior to disease onset in the babies who went on to develop NEC. And we chose a matched sample from control infants matched by gestational age, day of life, type of feeding, and mode of delivery. We analyzed the samples by 16S ribosomal RNA sequencing. This is a way of specifically looking at bacteria. We analyze a highly conserved sequence that's specific to bacteria and then compare those results to databases so that we can identify what microbes are present. Just looking at our initial data, we found that the NEC patients had lower diversity. So diversity is a comment about how many different organisms are there. And as mentioned previously, diversity is associated with a healthier community. So the control patients had higher diversity than the NEC patients. And then when we looked at the phylum level, the control patients only had about 40% of proteobacteria. This includes a lot of gram-negative organisms, and the balance was other phyla. Whereas the NEC patients had an increase in proteobacteria to over 90% of the community, with a very minor percentage being other phyla. One thing to note is that actually both of these percentages would be considered very high. In adult microbiome communities, proteobacteria should be less than 10% of the community. So even in the control patients, there was more proteobacteria than you would see in adults. We then looked at individual patients. So here, the top panel is babies with NEC, and the bottom panel is of control infants. The first 4 are the twins, and you can see the pairing for N1 and C1, and likewise to the N1, N4, and C4. 2 things to notice here. First of all, there's very few bars. What's labeled is the dominant genus, but you can see there's very few bars. These are very simple communities. An adult community should have hundreds of organisms, and we have just a handful for each patient. The other thing to notice is that for the NEC patients, there was no single organism that was associated with disease. However, each patient had a bloom to more than 50% of their community of a specific gamma proteobacteria. So all of these organisms, while they're different, they are all gamma proteobacteria. We did not see this in the control patients. They might have had a dominant organism in their community, but it was not a gamma proteobacteria, except for these 2 patients. So these 2 patients were chosen as controls initially based on their clinical presentation, but they did have more than 50% of Escherichia, which is a gamma proteobacteria, and both of these patients went on to develop NEC in the next 2 weeks. We then specifically looked at twin pairs. So when we matched C1 to N1, so we matched each patient with their specific twin, we saw that the control twins had decrease in proteobacteria compared to the NEC twin, increase in diversity compared to the NEC twin, and that the control patients separated from the NEC patients by disease state. They did not separate out by their twin status, and that wasn't necessarily predicted. When we looked at the complete cohort, we also saw separation. So what this is a PCA plot of a unifrac analysis. So each dot is a representation of a complete community in three-dimensional space, although this is a two-dimensional graph, obviously. And the closer the dots are together, the more similar the communities are to each other. And if there's separation, they're more distinct from each other. And you can see that the blue squares of the control patients do separate from the red dots of the NEC patients, except for these two. Again, these are the same two patients that we had chosen as controls who went on to develop NEC later. They actually clustered with the NEC patients. So this data told us two things. First of all, there isn't a specific pathogen associated with NEC. However, there is a microbial community associated with NEC, and it changes before NEC onset. So this led to another question. When do microbiome patterns change, and is it at a time point early enough to intervene? And for these studies, we looked again at our patients with and without NEC, and this time we analyzed the weekly fecal samples over time. And these pie charts are another plot where the samples that are closer in space represent those that are more similar to each other. And what you can see is that the initial samples of patients were similar to each other, but those who went on to develop NEC followed one trajectory, and those who remained healthy followed a different trajectory. And at this point in my research, I had a choice to make. I could either continue to follow the babies that went on to develop NEC, or I could follow those who remained healthy. And I actually chose to follow the ones who remained healthy because if you think about those risk factors, actually all of our babies are preterm and colonized with bacteria and are enterally fed and have altered intestinal blood flow. There must be something about those who don't go on to develop NEC that is perhaps protective. So then I analyzed the microbiome over time of just the babies that did not go on to develop NEC. And I compared this analysis to samples from a healthy full-term breastfed infant cared for at home. So this would be considered the gold standard of a healthy baby. You can see that for the full-term infant, weekly samples collected over about 8 weeks of life cluster pretty closely together. So we chose 8 weeks as sort of an average length of stay for a preterm infant. In that time frame, the infant cared for at home, the samples are pretty similar to each other. However, what we found was that there was a temporal progression of the preterm infant microbiome with a clustering around 2 weeks of life, another cluster around 3 to 5 weeks of life. And in these open white squares, it's only after 6 weeks of life that they begin to converge on the pattern of the full-term breastfed infant. So this suggests that there may be a developmental progression to the microbiome of the preterm infant that the full-term infant doesn't undergo. And this may make sense because the full-term infant doesn't have the same developmental hurdles that a preterm infant does. And this is consistent with what we know, that prematurity is not a disease state, prematurity is a developmental state. The preterm infant undergoes sort of a mistimed change in environment. So rather than completing development in the intrauterine environment, which is a relatively sterile environment where the placenta does all the work of oxygenation and providing nutrition, the preterm infant develops in the neonatal intensive care unit. And in this environment, host development coincides with microbiome development. And I wondered what role the microbiome could have in maturation. So we know there's many aspects of the preterm gut that are immature and may predispose to NEC. We know there's immaturity of cell surface glycoconjugates, which alters what microbes adhere to the intestinal surface. We know there's alteration in many aspects of innate immunity, decreased levels of gastric acid, decreased intestinal motility, decreased levels of secretory IgA, decreased levels of bacteriostatic proteins, intestinal trefoil factor, and human defensins. There's immaturity in intestinal mucus, which is intended to confine bacteria to the luminal space. Differences in terms of density and incorporation of secretory IgA. There's supposed to be tight junctions between cells, again, to limit crossing of bacteria, but this barrier is actually leaky in preterm infants, and this is thought to be a holdover from intrauterine life, where the amniotic fluid has large macromolecules that are important for development and important to allow them to cross. There's decreased numbers of B and T lymphocytes in preterm infants, and for the enterocyte itself, we know that there is increased NF-kappa B activation, which is associated with an increased inflammatory response to microbes, actually both pathogens and commensals. So I wonder, does the microbiome have a role in intestinal development? The problem, though, is it's actually easier to define a disease than sort of define development or a healthy outcome. So we needed something that we could measure to answer this question. And we chose to look at growth. So definitely studies have shown that babies who gain weight more rapidly while in the neonatal intensive care unit have improvement in long-term outcomes. Growth is sort of a summary metric that suggests adequate nutrition, resilience, resistance to infection. So this is what we chose to measure to look at the role of the microbiome in development. And for these studies, we used a nodobiotic mouse model. So nodobiotic just means known biota. And what we did was we studied germ-free mice and gave them specific microbiota. So the nodobiotic mouse model is used quite a bit in microbiome research, but I would argue it's particularly relevant to preterm infants, where there's a specific time point after which it is the normal situation where colonization rapidly increases. There's a prolonged period of observation for our preterm infants, or prolonged hospital course, a regulated diet, simple communities, and known follow-up. So for these studies, we took samples, fecal samples that contain the microbiome from infants with two different growth rates, high growth and low growth. And we all know that there's babies in the nursery. It doesn't matter how many calories you pour into them. It just seems that they can't grow. It seems there must be some other factor. We sequence samples from these babies, and going forward, H will signify high growth, L will signify low growth. And we took samples from the first 2 weeks of life, that first cluster that seemed to be significant, thinking that these initial microbes that encounter the naive intestine may have the greatest opportunity to have a significant effect on fundamental pathways. So 6 days old and 12 days old, both within the first 2 weeks of life. You can see in black that there were many common organisms. In red and blue are the organisms that were different. And you can see again, there actually weren't very many organisms. So these are simple communities. We took fecal lysates from the babies, transfaunated them or fed them by gavage to germ-free pregnant dams. And then the pups that were born, the litters acquired the microbiome of interest naturally from their mother through delivery and contact with the mother, which is sort of what happens to babies as well. We measured daily weight until weaning, and what we found was that high growth baby led to high growth pup, and low growth baby led to low growth pup. So now we have a model where we were able to recapitulate our clinical phenotype, but could also look closely at tissue. These pie charts just demonstrate that we were successful in our transformation. SPF is specific pathogen-free mouse pup. This is standard mouse colonization. You can see yellow and blue. This is Bacteroidetes and firmicutes. But you can see that all of our transfaunated mice, either the low growth dam, the low growth pup, the high growth dam, or the high growth pup, all have this purple color. This is proteobacteria, and similar to our babies, there's a significant percentage of proteobacteria that is not supposed to be in the mice. So this shows that we were successful in our transformation. And then we looked at the tissue. So as a measure of growth, we looked at intestinal length. This would be associated with nutrient absorption. You can see in the high growth, there was a difference in length compared to low growth. Over here, this pink staining is Ki67. It stains for cells that are proliferating, and you can see there's increased proliferation in the high growth compared to the low growth. Then we looked at markers of innate immune defense that we know are immature in preterm infants and are thought to be associated with NEC. So we looked at numbers of goblet cells. These are associated with producing mucus. Many more goblet cells in high growth than low growth, and it was statistically significantly different. There were differences in Paneth cells. So Paneth cells produce defensins, increased numbers in the high growth compared to low growth, and this was statistically significant. We looked at differences in tight junctions. So these were, again, the sealing between intestinal epithelial cells that are important for confining bacteria to the lumen and not allowing them to cross the intestinal barrier. We looked at two proteins, occludin and ZO1. You can see in the low growth, this is overall, and this is a blown up picture, they're these bright red dots, they're just scattered in the low growth, but you can see this nice continuous pattern in the high growth that suggests an intact continuous barrier for both of the tight junction proteins. And then we looked at inflammation. So you don't need to know what any of the words are, except this is low growth and high growth. What you should be able to see is that there's a lot of orange in the low growth, and orange signifies upregulation of genes associated with inflammation. So these are depictions of inflammatory response networks, and you can see that in low growth, there's much more upregulation of inflammation compared to high growth. And this was just in response to a difference in what we gave the mice in terms of microbiome. There was no secondary inflammatory stimulus. When we looked at specific inflammatory cytokines, blue is low growth, the purple color is high growth. You can see that for all the ones that we measured, there was increased production at these are samples from the ileum in the intestine, increased production of inflammatory cytokines just associated with the microbiome. And then we looked at NF-kappa B activation. So NF-kappa B is a transcription factor important for transcribing genes associated with inflammation in the immune system. You can see in low growth, there's much more activation of NF-kappa B, again, just in response to the microbiome compared to the high growth. And the other striking thing is that high growth is actually lower than SPF. So SPF is normal mouse colonization, H was actually lower. And so what this suggested to us is that the microbiome could do two things. The microbiome could increase your risk of having inflammation, or it could actually decrease your risk of having inflammation. And this would be associated with a maturation in your intestinal phenotype. So this again suggests that the microbiome could do beneficial things or harmful things. So this brings into question, how do you ensure then a beneficial microbiome? How do you change the microbiome? And I think this is where a lot of the interest in probiotics has come from, just this desire to improve the microbiome. So probiotics are living microorganisms in food and dietary supplements, which have beneficial health effects beyond their inherent nutritive value. But there are specific requirements. There should be benefit without toxic effects. The organism should remain viable during storage and use. They should retain the ability to colonize the gut, be capable of surviving in the gut, and have well understood properties. And the truth is, although probiotics have been used in clinical trials, we don't have a probiotic that meets these requirements. But the interest in probiotics has remained. There's several studies that have been done. I did not include the Preterm trial here, but these are representative. So each of the trials have shown a decrease in the incidence of NEC. However, there were differences in the probiotics that were used in these different studies. So it's not really clear what a recommendation would be. I think another concern with probiotics is that since we don't have a biomarker for who is most likely to develop NEC, probiotics have to be given to all preterm infants in order to decrease the incidence of NEC in some. We can't predict who's at higher risk. Therefore, there is a significant percentage of patients who would receive probiotics, who would be treated, but not at risk for NEC, because the incidence of NEC across the whole population is somewhat low, even if you pick a higher number like 10%. The Preterm trial had in their control group only 4%. So there's a lot of babies who would be treated, but not at risk for NEC. And that raises safety concerns. In addition, there are still open questions about what's the optimal probiotic organism, who do you give it to, dose, timing, length of treatment, mechanism of action is not understood, and there is not standardization of preparations. And there is a real infection risk. So there have been studies that have been published of bacteremia in preterm infants and probiotic therapy. There was a fatal case of gastrointestinal mucormycosis in an infant following use of ABC-Dophilus, which was used in a lot of the trials. And they did find that the probiotic itself was actually contaminated, and there are not specific guidelines for manufacture of probiotics. So the question is, should we give probiotics to a patient who is never going to get to develop NEC? And at this point, if I was in a smaller audience, I would ask, is there a time where we give an agent to a large population of people, of infants, to prevent disease in a few, and an intervention that we know has significant effects long-term and on the immune system? And I think the only time we do that is really with regards to vaccination. But what is the difference in the type of data that we have for vaccines versus probiotics? So I've heard it say, well, there's so much more data on probiotics compared to surfactant or cooling for HIE. Yet we are very comfortable with surfactant and cooling and much less confident about probiotics. But if you instead compare the data we have for probiotics to vaccine safety, for vaccines, safety and effectiveness is under constant study. They have to be extraordinarily safe. Safety testing begins as soon as a new vaccine is contemplated, continues until it is approved by the FDA, monitored indefinitely. And this has not been the case for probiotics in preterm infants. So then the question becomes, is there a different way to modify the microbiome? Is there a different way to end up with a beneficial microbiome besides just giving probiotics? And there are other ways. So this is a study on the role of human milk in extremely low birth weight infants. So in this study that looked at human milk intake in over 1,000 ELBW infants over several centers, when they looked at NEC or death between 14 days and discharge, they found that the hazard of NEC or death was decreased by 13% for every 100 mLs per kilo increase in human milk an infant received in first 14 days of life. Increasing the amount of human milk was associated with increased survival time free of NEC, and increasing proportion of human milk to total intake was also associated with decreased relative risk of NEC. So they concluded that a dose-related association of human milk feeding with a reduction of risk of NEC or death after the first 2 weeks of life exists among extremely preterm infants. Another way we could improve the microbiome is perhaps by limiting our use of antibiotics. So this is another study that looked at many infants, over 5,000 admitted to 19 centers, looking at NEC or death, NEC alone or death alone. What this study found was that many infants receive prolonged empirical therapy, which is defined as empirical antibiotic therapy for more than 5 days. This prolonged therapy was associated with increased odds of NEC or death. In fact, each empirical treatment day was associated with an increased risk. So these authors suggest that prolonged initial empirical antibiotic therapy may be associated with increased risk of NEC or death and should be used with caution, and speculated that choice of antibiotic duration is likely to affect the colonization of the neonatal intestine, which in animal models seems associated with the physiologic development of the intestine, immunologic development, absorbance of nutrients, and alterations and variation in any or all of these areas could increase the risk of NEC. So if you think now about modification of the early microbiome and NEC, we actually have 3 therapies. You could give probiotics, you could decrease number of days of antibiotics, or you could give actually any breast milk. All of these decrease the incidence of NEC. Now these 2 studies were from 2009. This is from the 2014 Cochrane review. And you can see here that the incidence of NEC in the control population is already much lower than previously. So there's also other things that we are doing in our overall neonatal care that decreases the risk of NEC separate from probiotics. And I put this up here as a caution. So these are multiple strains of a single organism, Lactobacillus reuteri, and this shows all of the cytokine profiles associated with different strains. And the main takeaway here is that they are all different. There's some decrease, there's some increase of the same cytokine. So consideration of host origin and functional differences at the subspecies level may have major impacts on probiotic strain selection and considerations of microbial ecology in mammalian species. So, it's very important when we think about probiotics to consider strain, and we are not to the point where we know strain level effects of these organisms. So what I would say that the facts are the microbiome is important in health and disease, but the term probiotics encompasses many different organisms, and we don't know what the probiotics are doing to the preterm infant microbiome. Under current protocols, the majority of preterm infants that would receive probiotics were never going to develop NEC, and we can't say that probiotics are without harm, particularly for the infants who are not going to develop NEC, and the studies have not followed these babies long term. And we have been here before. Things like oxygen and dexamethasone were used readily, and good things can be harmful at key developmental stages. I think we've all learned some caution. So I would suggest that rather than messing with the preterm infant microbiome with probiotics, we could think about protecting the preterm infant microbiome and recognize that probiotics have effects on the microbiome that we don't understand specifically with regard to impact on the overall microbial community. But what we can do instead is evaluate therapeutic options in light of effect on the microbiome, specifically decreasing antibiotic use, promoting exclusive breastfeeding. And these are potentially ways that we can protect the preterm infant, improve the microbiome, and induce intestinal maturation as well. So thank you, that's all I had today.
Thank you, Dr. Cloud, for really an eye-opening kind of presentation, and we could see that how complex and hard work is involved to reach to certain conclusions. And we will be taking the question and answers at the end of second talk. So you please be with us till then. And can you stop sharing your screen so that, so thanks a lot. We'll take a question answer, and I would ask everyone to put their question and answers into the question answer session. And I think we'll be having a lot of queries where people are already believing that probiotic is helping our babies, and they're already using it maybe. So we'll have a lot of questions, I think, and we would like you to be answering those. So let's move to the second talk, and I would introduce Dr. Hafiz to introduce our next speaker.
Hi, good evening, everyone. Thanks, Monica, for...
It's okay, yeah. So Dr. Hafiz is a consultant neonatologist in Cornish Hospital, and after getting his MD from a premier institute, PGI Chandigarh, he moved to UK, and he has done his subspecialty from UK, and he's involved in a lot of research work, and he's very, very keen on quality care, and he has recently published his paper on that and on the COVID. And it's my pleasure and honor to introduce Dr. Hafiz. All yours, Dr. Hafiz.
Thanks, Monica. Thanks for the kind introduction. When you're asked to introduce somebody like Professor Akhil, it's always a daunting task. So it's almost like preparing for an exam, just going through a CV. So I'll try to summarize as much as I can. So Akhil is an old friend. We have worked together in PGI Chandigarh, which I trained with Monica and Sridhar. So after graduating from Banaras Hindu University in India, he trained in pediatrics in one of the premier institutes, as Monica mentioned. And while we were there, we were contemporaries at that time. Akhil did stand out as an impressive intellect, and we all knew he was destined for greater things in academia, unlike most of us who are jobbing clinicians. So after his stint at PGI Chandigarh, he moved to the US, trained in Florida in pediatrics and neonatology, went on to take academic positions at University of Alabama, where he held a number of assistant professorships in immunology, gastroenterology, cellular and molecular biology, then moving to the University of Illinois at Chicago, again, South Florida, where he was a professor and held multiple co-chairs in molecular medicine, pediatrics, and public health. And finally, moving to Johns Hopkins as professor and chair of the neonatal service, where he is at the moment. So to talk about Akhil's research interest, it mainly focuses on gut inflammation and NEC. And recently, he's kind of working on the effect of transfusion on the immature gut and its correlation with NEC, which he'll be talking about more today. He has hundreds of publications to his name, a lot of first author, basic sciences publication, and has envious track record of kind of getting research grants. And he holds at least 8 patents for various research work he's done. And currently, Akhil has been working to bring together from the world over like-minded neonatologists like us to work and share our experiences in improving the outcomes of newborns and also understanding NEC better. So I can bask a little bit in reflected glory that I've worked with Akhil many years ago, and I too can say once walked with giants. So without much ado, Akhil, the floor is yours.
Today, I have been asked to talk about necrotizing enterocolitis and its relationship with red blood cell transfusions. And as all of us have talked about on numerous occasions, the question is this the devil? Or is it okay if we go by the existing standards? And over the next 45 minutes or so, I'll try to present it to the best of my knowledge. And if there are any questions, then we can certainly, give some time to discuss it further. I have nothing to disclose. There are no conflicts of interest. And these are the 3 things I will deal with. I'll try to spend the first fourth or so on the clinical evidence that we have, which has resulted into the clinical controversy that we have, that each one of us has when we try to treat an infant with a red blood cell transfusion. Over the next 30 minutes or so, I'll try to present the laboratory data that we have acquired over the past few years. And in the last 20% of the talk is exactly where do we stand? Have we been able to resolve this controversy to some extent? So the first question is, why did we start asking this question? What is the clinical evidence? And in the next few slides, I'll try to summarize as to what we know. And to start with, I think the best way is to start by looking at the meta-analysis of pooled data. Mohammed and Shah from Canada first reviewed 11 observational studies. So these are all observational. And the data showed increased odds of developing necrotizing enterocolitis within 48 hours after receiving a blood transfusion. In meta-analysis of 5 studies that they found to be eligible for inclusion in this statistical analysis, they noted increased odds, almost 4-fold, which were statistically significant. In another study, Tao and his co-workers reviewed 7 case-control studies, and there were 480 cases of transfusion and a fairly large number of controls. The odds ratio was still increased in the next 48 hours, and they found it to be almost 4-fold. Most studies with negative effects of transfusion were retrospective, observational, and were limited by a small number of subjects that were looked at. Several studies showed conflicting results, adding to the controversy. And more recently, there was another meta-analysis where Garg, who is at University of Mississippi, he analyzed 17 studies, so a much larger number of these studies, and they did not find any increase in the risk of necrotizing. Another meta-analysis by Rai and his colleagues with 10 studies, they actually found a 45% reduction in the odds of developing necrotizing enterocolitis after a recent transfusion. So fairly controversial. In other studies, Sharma and his co-workers, they compared prospectively, they compared 42 cases who developed necrotizing enterocolitis versus matched controls at 3 centers. It was a small n, but they saw no association of transfusions with necrotizing enterocolitis in the following 48 hours or in the 7 days after this transfusion. In another study, El Abiyad and his co-workers, they reviewed 174 cases with NEC. 116 had received transfusions. The transfusions were not associated with necrotizing enterocolitis in babies who were born with a birth weight less than 1,000 grams, but they remained a risk factor in those born between 1,000 and 1,500 grams. In another study, Ravi Patel from Emory University in Atlanta, he evaluated nearly 600 very low birth weight infants it was a secondary but prospective, multi-center but observational cohort. They noted necrotizing enterocolitis in 44 infants, which was about 7%, it's representative. And at 8 weeks of chronological age, the incidence of necrotizing enterocolitis was higher in infants who had received a transfusion versus those who had not. The incidence of NEC was 9.9% versus 4.6%. So it was a difference. It was a significant difference. They associated necrotizing enterocolitis with severe anemia. The hazard ratio was almost 6. It was highly significant, but they did not find an association with RBC transfusions. So if we look at the clinical part, it looks fairly confusing. Then there are other clinical studies. When we look beyond red cell transfusions, you can look at recombinant erythropoietin or one of its longer half-life derivative, darbepoetin, which may reduce the need for transfusions, and they found a trend towards protection against necrotizing enterocolitis. In another study, Olson and Aher, they studied nearly 3,600 infants. These were all low birth weight, and one group received early, like before 8 days, erythropoietin and darbepoetin, and they compared this group of infants with those who did not receive EPO or darbepoetin, but had received placebo. Early administration of erythropoietin reduced the need for red cell transfusions and donor exposures. And in a sub-analysis of 15 studies reporting 2639 infants, a fairly good number, the administration of erythropoietin or darbepoetin reduced the NEC incidence of NEC. The risk ratio was 0.69, and the number to treat to benefit was 33, so not too bad. So we developed an animal model in our lab, and we took 4 groups of animals. The first group was animals which were just observed. This was a group of naive mathematical controls, and these animals were just observed. And we observed these animals for 13 days. The second group of animals who were otherwise normal, but had received a blood transfuser exposure. And this was given on postnatal day 11. The third group was a group of mouse pups which had been exposed to venous phlebotomy on postnatal days 2, 4, 6, 8, and 10. And by doing that, we rendered these mouse pups to develop severe anemia. That is, their blood hematocrit was between 20 and 24%. So these were severely anemic mouse pups. And in the fourth group, these severely anemic received a blood transfusion, which was given after the 4th day in an attempt to correct the severity of anemia and to simulate what we see in the clinical situation. And what we found was that in mouse pups, which were in the naive control group, in the transfusion control group, or in the anemic control group, the intestine remained normal. But in the 4th group of anemic mice, which received a transfusion, they developed intestinal injury in the distal ileum, in the cecal area, and in parts of the large intestine. We looked at these samples under the microscope, and what you see is that the naive control were normal. This is the small intestine. This is the large intestine. Mouse pups, which just received a transfusion, were also normal. So no injury in small or large intestine. Then in mouse pups, which just had anemia, had some degree of inflammation, and I'll show that in subsequent slides, in both the small intestine or the large intestine, but there was no mucosal injury. There was inflammation, but no damage. And in the 4th group, these were anemic mice which had been treated with red cell transfusions. Those mice developed histopathological evidence of injury, both in small intestine and in the large intestine. And we often describe intestinal injury in 4 grades. In grade 1, it is only mucosal injury. In grade 2, you have submucosal injury. In grade 3, you have involvement of the muscularis layer. And in the grade 4, there is a transmural injury. So these are grades 1, 2, 3, and 4. And what you see is that there was intestinal injury only in mouse pups that were anemic and had subsequently received a RBC transfusion. This is measurement of fatty acid binding protein number 2. It is a protein that is released from the intestine during intestinal injury. This can be measured in the bloodstream and can be measured as nanograms per milliliter. And you see that this FABP2 is increased only in the anemic transfused mice, meaning this gives further evidence to what we see in histopathology in this slide. And again, to emphasize that the histopathological evidence of injury was only in this 4th group. When we looked further, I said we noticed some inflammation in anemic mice. So we started looking at the number of leukocytes just under the microscope. And what we found was that there was no inflammation, obviously, in untreated mice. Surprisingly, there was also no inflammation in normal mice which had just received a red blood cell transfusion. But there was inflammation in anemic mice. There was increased leukocyte infiltration, but did not have any obvious mucosal damage. But the 4th group, which had mucosal damage, also had inflammation. So inflammation was seen in anemic, but with no mucosal damage, and anemic transfused mice, which also had mucosal damage. So these two had inflammation. This came as a surprise. When we look at histopathological sections from these two groups, what came as a surprise was that the inflammatory cells had the appearance of macrophages. When we look at macrophages in histopathological slides, they're often nuclei with a single indentation, like a bilobed nucleus, and with a large amount of cytoplasm, which appears pinkish, eosinophilic. And you find a large number of monocyte macrophage-like cells in these tissue sections. So we started looking at these monocytes macrophages. And this may appear complex at the first look, but I'll explain everything. So we first looked at mouse pups, which were in the naive controls group. That means these mice or mouse pups were normal. we hadn't done anything to these bulbs. And when we look at these cells by flow cytometry, then these cells, each dot represents a cell, then these dots actually reflect myeloid cells or white blood cells. And these have this antigen called CD11b. We then took these cells and looked for a macrophage marker. And these three groups, these are called the resident macrophages. This group represents the newly recruited macrophages, which come from blood monocytes. And these are non-macrophage cells. This is towards the negative side. So this does not express any marker for macrophages. This is naive control, mice which had no intervention. The second group is normal mice, which had just been treated with a blood transfusion. And when you looked at this, and we just took the same subgroup of cells, you found only the same subgroup. You found that these are F480 positive macrophages, but nothing else. So transfusions by themselves was not poison. The third group was just anemic mice. These had not been transfused, but you see that the number of white cells had increased. And now when you analyze this population on the right side was still there. But now you had an additional population where these white cells came in. And these are typically called F480, which is a macrophage marker, but F480 mid. And in immunological studies, these cells are typically considered to be monocytes. The fourth group is, and you see the increase in number, these are now 36%. And when you transfuse these anemic mice, then you still found this upper population to be high, and you still saw a further increase in the total number of macrophages from 36 to 49, and you still had this middle population. These newly recruited, so these are macrophages, F480 is a macrophage marker. These are macrophages were the cells which were originally in anemic cells. And these red cells they comprised of the original cells and also the cells which came in. These are derived from newly recruited monocytes. And you see that these guys in the red bars, they expressed all kinds of inflammatory markers. So the characteristics of these cells had changed. These cells now became activated and they became inflammatory. This is the fold change in gene expression. So if you see, the higher bars are the ones which are inflammatory. Then we wanted to investigate these further. On the first look, these slides may look complex, but they're not. So these mice are naive control, and the white blood cells are only towards the right. These are the cells which are native cells. They were there in the intestine always, in normal mice. Then there is a drug called clodronate. If we administer clodronate, then we largely remove these cells from the mice. These specifically deplete this cell population. In the anemic transfused mice, you see that the number of cells in this column and in this section have appeared. This has increased. These are the newly recruited cells. And then when we give clodronate, which is the chemical that depletes a mouse of this middle population, these macro-inflammatory macrophages, you see that we achieved our ambition, that these cells were decreased, but this new population disappeared. We measured this, and you see this red line is the transfusion control. The blue line below that is the anemic control. So nothing happened there. But in clodronate control, there were no macrophages. And when we give clodronate and anemic transfused mice, you were able to reduce mortality. Here I have mentioned as physical distress, not mortality, because by rules, in mice that develop physical distress, we take that as an early indicator of intestinal injury and we sacrifice these animals. And you see that in the mice which had been treated with clodronate, those who did not need to be sacrificed, only the anemic transfused mice, they needed to be sacrificed or they died spontaneously. And this is showing the same information by a different way, that when you treated the anemic transfused mice with clodronate, the grade of injury was significantly lower. So macrophages were the cells which caused intestinal injury. We wanted to confirm that further. And this slide might appear complicated, but again, I'll walk you through it. It is not that complicated. One of the markers of macrophages is the colony stimulating factor receptor 1 gene. This is expressed exclusively on monocyte precursors or monocyte colony stimulating factor receptor. In the exon 21, exons are the sections of a gene which eventually help in production of a protein. We modified this exon and we inserted a stop codon. And this is an internal ribosomal entry site. Don't worry about it. This is a diphtheria toxin receptor, and this is a red marker which we inserted. So these mice are genetically modified. Just let's go with that. When we cross this mouse with another mouse which expresses this particular genetic sequence, then you selectively cut out this stop codon. And these three genes start becoming expressed. And this is what I'm saying here. When you do that, then you modify your mouse. So these are the mice which were genetically modified, but these genes become activated only when you administer diphtheria toxin. And in these mice, when they're anemic and have been transfused, you see the two populations. When you administer diphtheria toxin, you modify these mice genetically and you remove the middle population of inflammatory macrophages. When you do that, you reduce the mortality. So this complex looking slide, all it shows is that when you remove this middle population, then you reduce mortality. So the mortality is occurring from newly recruited monocyte-derived macrophages. You see this by another means. We have immunostained these intestinal sections. The green dots are the native macrophages. The red cells they're enlarged, elongated, meaning thereby these were activated. These are monocyte-derived macrophages. And these red cells are seen only in anemic or anemic transfused. So these cells were recruited. You see that these cells were recruited in anemic cells, but transfusions were required to activate these cells so that the tissue got damaged. You see these cells that were drawn out of these animals. In anemic, you saw these monocyte-derived macrophages. But when you transfuse these, you see all these pseudopodia, which means that these cells actually became active. This is another way to show activation, that these cells which look green, they also started expressing this red cell, which is a marker of activation. In the anemic cells, the cells were there. You see these green dots. These were all macrophages, but they did not express red color. So that means these cells were not activated. But in anemic transfused, it was the transfusion which activated these cells, and now they start showing both red and green, which is seen as yellow. We showed that by different methods. we isolated these cells, and NF-kappa B is a marker of activation. And by various methods, we showed, recombining everything, that it is the red cells which activate these cells, these monocyte-derived macrophages. We showed that further, that it was the heme component in the transfused cells, transfused red cells, which were causing this damage. We again showed how, and it was that the hemoglobin that was derived from these transfused red blood cells, they activate toll-like receptor 4, the same receptor that is activated by bacterial endotoxin. So all of us know that the bacterial flora, that is actually what activates the inflammatory pathways. But what we found in this study is that it is the hemoglobin that is released from stored red cells which activate the same receptor that is activated by bacterial endotoxin and causes intestinal injury. Another way to say that, this is further evidence that the red cell transfusions activate toll-like receptor 4, the receptor for bacterial endotoxin, and that causes injury. This evidence is from mice that have been genetically altered so that they don't express the toll-like receptor 4. And when we did that, then there was no injury. Another way to show that, the green shadows show activation of NF-kappa B, which is an inflammatory marker. And when we block NF-kappa B by giving nanoparticles, then the injury goes away. Then we protect these mice. Just multiple ways to show that the pathway we were looking at is real. And again, this is something that is clinically relevant to you and me, that this transfusion-induced injury is much more severe in pups with severe anemia, but was lesser in those with moderate anemia. And I'll come back to this. Then it is the duration that you spend in severe anemia. So if you allowed these mouse pups to remain anemic for 24 hours, injury is more. If you transfuse sooner, injury is less. You could also ask that, hey, if I wash my red cells, would I reduce the level of injury? And indeed, if you removed the white cells and you washed your red cells, the red cells you transfused, the injury was decreased. Does the duration affect? The conventional intelligence is it should, but we did not find significant evidence. So the consideration that you reduce the number of donors might still be valid. So you take red blood cell from one donor and use the same donor to transfuse again and again. As long as your red cells are not damaged, you may be okay, but it needs further study. Does the number of transfusions affect? And yes, indeed. If you give more than one transfusion, your risk is higher. And this is being shown in a different way. This is the number of transfusions, and this is the grade of injury. So the severity of injury also increased as the number of transfusions increased. Now I go to the third part, that I have clinical evidence which was uncertain. Then we looked at the animal model, which looked convincing. So where do we go from there as clinicians? One is transfusions by themselves were not injurious. So maybe the use of the word transfusion-associated NEC needs to be modified. And maybe it needs to be called transfusion and anemia-associated NEC to emphasize the importance of severe anemia. The second thing is that our current evidence favors minimizing the exposure of premature evidence, potentially at high risk of NEC, to prolonged and severe anemia. At present, there are 4 sets of national guidelines that are available. And the risk of transfusion seems to increase with chronological age. These guidelines are from Britain. They're from Australia, Canada, and one another country in Europe. But it is the chronological age which is an important consideration. We do need to identify safe hematocrit or hemoglobin thresholds for RBC transfusions. How anemic can we let them go? We also need to evaluate, once again, the potential benefits of erythropoietin or darbepoietin, at least in specific subsets, like babies with congenital cardiac defects, if not in every at-risk infant. Only a minority of severely anemic premature infants develop NEC, and so there is a need to identify genetic risk factors or clinical risk factors, because not everyone who is severely anemic develops necrotizing enterocolitis after receiving a red cell transfusion. Considering the typically delayed onset of anemia of prematurity after birth, studies are needed to identify postnatal periods of increased vulnerability for more intensive monitoring. You cannot put multiple monitors on every premature baby, because then you're reducing the opportunities to improve maternal-infant bonding. If the baby has 15 monitors attached to him or her, how do you do that? But if you define a chronological period in which the baby is at higher risk, then it may be possible. It may be a smaller cost. Then we need to identify infants with ongoing subtle subclinical hemolysis. These infants may have anemia, but they also might have activation of monocytes and macrophages and the need for repeated transfusions. Everything we have found to be associated with transfusion associated with anemia and NEC. Again, to show it pictorially, it is the free hemoglobin which might activate toll-like receptors in the intestine. It is the free hemoglobin that is important. It is not the red cells that may be important. And this free hemoglobin might activate blood monocytes or intestinal macrophages, and these two sets of blood cells activate and damage the intestine. Now, what about vascular autoregulation in the intestine? So splanchnic vascular autoregulation needs further evaluation. And suddenly we're in a position where handheld or pocket-held ultrasound machines might enhance the feasibility of this. Association of transfusion and anemia-associated NEC with stimuli that increased intestinal oxygen consumption, like feeding, might need evaluation yet again. Transfusion thresholds of hematocrit or hemoglobin are still unclear. Animal models suggest 24%. But remember, mice don't have fetal and adult hemoglobin. So some human studies are still needed. And some human studies that I did with collaborators in St. Louis, they actually suggest that the intestine may be at risk of 24%, but the brain might actually be at risk at 28%. So the story is not done. We need more. We need to view red cell transfusion thresholds as a range, not exact levels of hematocrit or hemoglobin, because the corrected gestational age, altitudes of residence, like my collaborators in Utah, which is at a higher altitude, they may be at different comorbidities and the functional status of the microcirculation, because you might have a small for gestational age baby who might have altered microcirculation. The rest may be completely different. Then murine studies do not show harm in transfusions with red cells stored for short periods. These findings are at variance from what we know currently, that RBC storage is a bad thing. We need to look at it yet again. Murine studies show no harm in transfusions with short periods. And these, again, I'm repeating this to emphasize this, that these findings are at variance with existing evidence from human studies. Need to look at it again. The concept of anemia in premature infants needs evaluation. Fetal and adult hemoglobins have different oxygen carrying capacities, and so low hematocrit levels in an infant who has previously received red cell transfusions from adult donors may have completely different physiological implications than another infant who is not. The advantages and disadvantages of washing red cells needs another reevaluation. Washing may reduce the half-life of transfused red cells, but it may remove free hemoglobin and the red cell contents to reduce the risk of NEC. And that's where we need to look at mouse and human studies very, very carefully, because mice are not men. Severe anemia may also affect the brain. The intestine is not everything, and follow-up studies may need to carefully examine the outcomes of these infants in comparison to those with NEC related to other predisposing factors. We also need further study to confirm that transfusion-associated NEC in human infants is more likely to involve the ileocecal region. And when we consider that transfusion and anemia-associated NEC could be delayed, the colonic region needs to be very carefully assessed. The relationship between enteral feedings and transfusion and anemia-associated NEC needs to be carefully evaluated. Even though observational in design, there is a beautiful study going on in the United Kingdom. It's called the WHEAT study, withholding enteral feeds around packed red cell transfusions. And these studies like this may yield very important insights. And nutritional interventions, pasteurized human donor milk, enteral substitution of lactoferrin or L-arginine, and preventive application of multiple strain antibiotics. I think these need to be examined very carefully with sufficient monitoring, such as with ultrasound. This is where I'll stop. I wanted to thank some of my collaborators and colleagues. They're here, and this is here where I'll stop. Thank you so much for giving me this opportunity.
First of all, I have to say it's fascinating work both of you have presented, and we frontline clinicians actually owe a great gratitude of debt to basic science researchers like you to kind of further our understanding of diseases which we grapple day in and day out. And slowly but steadily we are getting that. The evidence is being generated by the likes of you, and thank you for that. So we'll start with a question for Erika from the audience. 1 second, let me. There's a question from Dr. Prakash Mechnikoff about the microbiome. So spontaneous intestinal perforation is quite different from NEC. Does the microbiome have any role in SIP, or is it completely unrelated? Have you found any evidence to that?
That isn't a patient population that has been well studied. My guess, generally we see SIP much earlier in time. So I think SIP is much more likely to be a vascular condition. So I don't think the microbiome will have had as large a role to play in that disease, but it has not been well studied.
Okay. One more question for you, Erika, which says, because there are concerns which you raised about the safety of probiotics. So saying, should we be looking at prebiotics rather than probiotics? Is that the way forward? And how much experience do you have with that?
I definitely think prebiotics are important and likely safer than probiotics. I think the key thing with any of these things is that we should understand the mechanism of what we're giving. So, you know, obviously the best source of prebiotics is going to be mother's own milk. I mean, I would actually argue that mother's own milk is going to be better for this than donor milk. And that is a great source of oligosaccharides and other prebiotics. But the advantage to prebiotics is that they are more specifically studied. I think one of the concerns with probiotics is it's kind of used as a general term. It's interesting, you know, we would never just use any old antibiotic or any old vaccine. These are categories, but people sort of think probiotics, you can just use whatever you have and they're all the same. And that's where I think the danger is.
One last question for Erika before moving on to Akhil. Sorry, this is for Akhil. A couple of people have asked this question. So translating your work into the clinical arena, you talked about the possibility of increased oxygen consumption during transfusions, like with feeding. In your practice, both of you, Erika and Akhil, do you stop feeds during transfusion?
Erika, do you want to go first?
The practice of our nursery is to stop the feeds around the time of transfusion. You know, there have been studies done, I don't know if they were in the ones that you presented, Akhil, where there was still significant, quote unquote, transfusion related NEC, even when feeds were held. But, I think that's an abundance of caution, not necessarily, clearly supported. There are situations where we have not been able to, for instance, get another point of access to be able to deliver IV fluids during the transfusion, because by doing this, we require 2 lines, and in those situations, occasionally we'll feed, but the general practice is not.
How about you, Akhil?
This is a great question, because, you deal with this situation both as a human being and as a physician. As a human being, all of us find it extremely hard to see a baby crying with hunger. In terms of science, there are some studies definitely which show that withholding feeds during transfusions might make it safer. The quality of science is soft because we haven't been able to decide that if we stop feeds for how long. Because if you look at sonographic studies, then the reversal of feeding or anemia or transfusion-induced alterations in splenic blood flow, those are variable, but probably longer than any of us have stopped feeds. So in many studies, we have stopped feeds for the duration of the transfusion. But as you saw, it's not the transfusion which is the devil. It may be a combination of anemia and transfusion. And the severity of anemia has also varied between studies. The other issue is that the effect of transfusions in the causation of anemia is only in a minority. So I do not think we are ready for it yet. I think this question needs to be looked at all over again in a greater degree of detail, because all of us go through this psychological, philosophical dilemma as to, do I know enough to stop feedings from this crying baby?
So do you or do you not stop feeds? What's the practice on your clinical unit?
We do.
Okay.
The transfusion, it's a very soft decision.
Okay. Sure. Thank you. I'll hand over to Monica to carry on with the question.
Same
Thanks.
question, like same thing, but a little different question is that Dr. Kishore Sangvi has asked, because you said about the threshold of transfusion, what should we balance? Because if we go down, we are seeing NEC. If we go up, we see ROP and BPD. So what do you strike balance? You gave the answer about the animals that you put a cutoff of 24, but what about the humans? What is the cutoff in your unit, and how do you balance it out?
So we're still using a cutoff of 24, because most criteria, most guidelines for transfusion have been restrictive. And increasingly, the studies have showed that by reducing the number of transfusions, you reduce the exposure to donors. And in the major outcome measures, there has not been a difference. Where the doubt has begun to come in is from the studies that we have done in collaboration with St. Louis University. In the gut is gut. What we are beginning to find is the difference in the neurological outcome. And I think that may be something that needs to be followed. The difficulty is we're still not sure as to what is the best experimental setup, because mice do not have fetal and adult hemoglobins. So this is something we might have to do using a porcine model. We're still thinking about it. We are not there yet. But what I'm meaning, the short summary for that long answer is that NEC or intestinal outcomes might not be the endpoint to answer this question. We might have to look at other organs at the same time.
Okay, so 24 you mean is your unit cutoff, right?
Yes, because that's how we've been defining severe anemia. And the occurrence of NEC, fortunately, is still in a minority. And it seems that there are 2 subsets of infants with NEC. One subset may be practice related, because as all of us have started, instituting certain practice improvement measures, we have seen reductions, important reductions in the incidence of NEC. But then the second subset might be biological. So even in the best of practices, the incidence has sort of plateaued at 3%. So that's where the confusion is. The number of infants affected is small, and the impact of practice changes seems to be unkind.
Okay, thank you. Dr. Arika, Dr. Amir Ahmed is asking, like, is there any difference in the microbiome of a baby born vaginally and cesarean section? And would you change your practice, like if the baby is born vaginally or in cesarean, while you think about giving probiotics?
There's certainly been data in full-term infants that shows difference between vaginal and cesarean section delivered infants. The studies of preterm infants, however, have shown that the primary driver of the microbiome is really just gestational age. And so that was a bit of a surprise, whether that is because the balance, so we definitely have more preterm infants that are delivered by cesarean section is affecting the data, or whether the practice in the neonatal intensive care unit over time is such that it very quickly drowns out the effect of vaginal delivery is not clear. But we just recently published a paper that actually showed that the first 48 hours of antibiotics also doesn't affect the long-term microbiome or outcome. So I think that the overall course of a preterm infant changes so rapidly after delivery, that is not a clear difference for our patient population.
Okay. And Dr. Arif wants to ask you that does use of Prolacta, that is humanized HMF, would reduce the risk of NEC because it's coming up in a big way and it looks quite expensive, and should we go for it or not? Yeah.
So it has been shown to decrease the incidence of NEC. It's actually been associated with the transfusion-related NEC as well. There was some comment, I think, that there was a difference in bovine feeding associated with transfusion-related NEC, but it is very expensive, as you know. So we are not using it.
I would agree with that.
Just because of expense, or it's because of some other reason?
The expense is probably the primary reason that we aren't using it, but I think there's other ways to modify the microbiome. You know, at that point, that I still am a strong proponent of own mother's milk. So even that that formulation is not going to be the milk of the mother of that baby. There are pasteurization procedures, so you are losing much of the benefit. There's certainly, in terms of informing the microbiome, there are organisms that are passed from mother to baby. So you're losing a lot of that. You're losing, in the pasteurization process, a lot of the benefits of own mother's milk. So I think really, if you're going to consider resources and costs, ways to improve own mother's milk is still going to be better.
May I add a sentence to that? So this was a question that came up from UAE recently. One of the physicians had asked, what did I think about it? And I think because the advantages or disadvantages are very relative, some of these might have to be considered by what is the existing incidence of NEC in your unit? Because if the incidence is, say, 10%, then the advantages would become more evident. In our units, the incidence is lower. So the disadvantages become more prominent. I don't know, Erica, what do you think?
I think that is an important point. I think the other issue, though, is even if it's 10% of infants developing NEC, that's still a large group that isn't developing NEC. I think one of the issues that neither of us mentioned in our presentations, but it is relevant, what we could really use is some biomarkers or some ways to predict the patient population at highest risk for NEC. And to those patients, perhaps you could consider more of these interventions. But at this point, where the only risk factor that we can identify is prematurity in general, so we have to administer whatever therapy to all the premature infants, that makes it difficult.
Yeah, Hafiz, you want to ask? Because I'll ask a question. Yeah.
Yeah, I have one question for myself. Just bringing both your research streams together. Is there, because transfusion seems to affect, have some sort of effect in relation to anemia and NEC, so does the microbiome. So Erika, in your studies on the microbiome of children who were affected by NEC, was there a difference, significant difference in the group which was transfusion associated? Have you looked at that? And the non-transfusion associated NEC, which just happens out of the blue?
That's a very interesting question. We actually have not looked at that. I don't know, Keel, have you looked at that or seen literature?
We haven't either. And I also, philosophically, since we started looking at these studies, both in humans and in mice, interestingly, it is the effect of anemia, clinical or subclinical, which is becoming more important to us. NEC is only the tip of the iceberg, because in the studies with St. Louis, once we started looking at cerebral outcomes, then you start worrying that in the attempts to reduce the exposure to red cells, do we have the full story yet? And maybe we don't. So is it early initiation of darbepoetin or something like that? Because as Erika mentioned, all of us worry about exposure to red cells from other people, right? Or milk from other people. So I think the answers are yet to come, but I think these have to be very carefully considered.
Yeah.
Monica, any more questions?
Yeah, Dr. Saad is asking, Akhil, can this NEC be just a type of a graft versus host reaction when we are seeing it after transfusion?
That's a very interesting question, a very different way of thinking. The reason we have not thought on those lines is because the inflammatory response is primarily myeloid. The way we have thought of graft versus host reactions is as involving lymphocytes. And here, at least in the studies that we have done, primarily the inflammatory response is myeloid. And that's why we haven't gone in that direction. Now, would we identify a subset of myeloid cells which are activated by antigens? That is possible. There are some studies which are showing that there are certain myeloid cells which might not be innate immune cells. And if that happens, we might have to think differently. But right now, we don't think so.
Okay. Dr. Erika, Dr. Dua is asking, would you recommend probiotic in a preterm infant who is post-NEC now and a baby who is short bowel syndrome?
So that's interesting research that's going on. I do think that probiotics can have a role in growth, but we're then left with the same issue. We still would need to identify a specific probiotic. So, probiotics is not a single therapy. You would still have to identify a specific agent, a specific mechanism to understand it well. So I do think the microbiome can have a role in regeneration. We have to understand more before we recommend a product.
Another question for you, Dr. Heba. I mean, they're thinking really ahead. I think you made us all think like that, So they're asking, do you think that dysbiosis happens during intrauterine life? And so if we give, these probiotics to these pregnant moms, the prebiotics to this pregnant mom, they can help to decrease the incidence of NEC?
So I think that's a great idea. I am actually personally not necessarily a believer in the intrauterine microbiome. There's been conflicting literature around that, about whether or not there really is an intrauterine microbiome. However, the initial microbiome to the baby does come from the mother. We know that preterm delivery itself is triggered by alterations in the microbiome. Sure, we could eradicate NEC if we could get rid of preterm delivery. So maybe we could give probiotics to mothers for whom we are not as concerned about their immune system and some immaturity there. And perhaps there could be a role in decreasing the incidence of prematurity itself. But I do think that administering the probiotics, again, if we knew what agent to give, would be a way to perhaps alter the initial microbiome that babies are colonized with in ways that might have an effect.
Thank you.
We'll probably take one last question and then wind up. So this is in relation to feeding. One of the risk factors of NEC has always been growth restriction and reduction in the umbilical artery flow antenatally. So I know the ADEPT trial from the UK didn't show much of a difference in terms of delayed feeding if you had absent end-diastolic flow. And also the speed of feeding, the SIFT trial showed even rapid feeding versus slow feeding, the increments didn't show much of a difference. So what is your policy, both on your unit, of feeding small for gestational age infants or preterm infants with restricted umbilical artery flow on antenatal dog blood?
Erica, do you want to go first, or?
We are cautious, but we still advance by 20 cc's per kilo per day. We don't, I mean, the meta-analyses have shown for sort of general preterm populations, anything up to 40 cc's per kilo per day didn't seem to have an effect. So we aren't feeding to that extent in general preterm infants, but we also aren't decreasing the rate for those who are specifically small for gestational age either.
Okay. Do you delay starting the feeds at all?
No, if the baby is otherwise appearing well, we do not. We might delay the start of feeds if the baby was specifically unstable after birth, but not based on earlier Dopplers.
Okay.
So the way I have begun to think about this issue.
Sorry, Hafiz, there are very questions. Yeah.
Sorry, can't hear you, Monica.
No, Akhil want to say something. Yeah.
Yeah, sure.
So the way I have begun to think of feedings has changed. After looking at the mouse model for the transfusion and anemia-induced NEC, I have started thinking of transfusions or feeding or hypothermia or congenital heart disease more as the triggers. It is the previously inflamed intestine which might be affected by a trigger. So I think we may need to look at these problems in two components. One is as to what is the previous status of the intestine. And second is we need to find ways to monitor infants who are at higher risk more carefully. And to me, the most exciting thing that's coming up is the handheld ultrasound. I think 5 years from now, I think I will be doing those ultrasounds. I will be following the splank neck blood flow. I will be looking for evidence for mucosal thickening and so on. Right now, as Erica said, that's exactly what we're doing. We feed carefully, we keep watching those infants. We fail sometimes, and those babies develop.
Yeah, I think there are a couple more pressing questions which Monica wanted to ask. Monica.
Yeah, I mean, it was interesting question. So I wanted to ask, like, is it only RBC or other blood products have the same kind of a trigger? And you would be cautious on those as well. And another question is there any really role of antibiotic in the neck? Because neck is like going inflammation because of other reasons. So that's for Dr. Arif. Yeah.
Do you want to go? Yeah. So we have looked at obviously the role of red cells. In our models or in our review of clinical literature, platelets seem to augment the severity of intestinal injury, but do not trigger that. So, and the number of white blood cells in the transfused aliquots of blood does not seem to be large enough to cause injury, because at least in the United States, most blood banks are increasingly using precautions to reduce the number of white cells. There still are some lymphocytes and monocytes, but considering the fact that transfused blood did not cause NEC in non-anemic infants, is an important consideration. Transfused blood in infants in periods before, like in earlier weeks, did not cause NEC. So increasingly, the way we have begun to think is that whether there is a substrate and blood is an inducer. So that is the change. So our goal will be to look at the substrate increasing.
What about the second question, antibiotic and NEC? I mean, do we really need to give antibiotic in NEC? Because is it really infectious?
That is a great question. So there have been a couple of studies which have shown protection. But so will antibiotics protect against NEC? Possibly. Do we want to give antibiotics? Possibly not. Because then what will I give when a baby actually becomes septic? So that is a question, because the number needed to treat with antibiotics to prevent one case of NEC will be fairly large. And you all worry that will it lead to increased antibiotic resistance, and will I have anything to give to a baby who's critically ill?
I think there were early studies done on that, and that's exactly what they showed, that there was a decreased incidence of NEC and an increased risk of antibiotic resistance. So it's not done routinely. I didn't know if the question was about giving antibiotics once a baby was already sick. In that case, I think part of the issue is you have a damaged intestine and risk at that point for bacterial translocation. So even if there isn't a positive blood culture, for sure there is bacteremia that you're not capturing, and the antibiotics have a role at that point. I don't actually think it has a role. Once we have clinical symptoms, the process is very far along. I don't actually think at that point the purpose of the antibiotics is to try to mitigate the actual intestinal process. The inflammation has started. However, you can decrease the risk of further compromise from bacteria that are translocating across that damaged intestine.
Okay, I think we've covered most of the questions asked, and probably it's getting late now for everyone east of us and slightly for the people here. So we'll wind up here. Sridhar, do you want to give the parting remarks?
I think there is some.
Sorry, Monica, you mentioned something?
Yeah.
I'll catch up on the video. Thank you, Akhil.
I want to stop here. I just want to ask one last question, because I think asked, like, irradiated blood, would it help in decreasing the NEC? That would be the last question.
It's a great question. There are some clinical studies which suggest that yes, there is a reduction, but I think we need to study it very carefully in the mouse models, because the number of cells which are being, white cells which are being transfused inadvertently in red cell transfusions is small. So if I were to make a guess, then probably not a major difference. I don't know, Erica, how you think about it.
I agree with you. I don't think it's going to make a significant difference, but we don't know. We don't have information to answer that one or the other.
So, Sridhar, do you want
I
to?
have many more questions. I think I'll post them to you, and you guys can answer, and I will post back to the people who are asking them, because there are, I think, 30 more questions and time is limit, so we'll do that later. So Sridhar, can you do a closing remark to thank the speakers? Yeah.
Thank you. I mean, apologies that I couldn't join earlier, but Akhil is a great friend of mine, and first time I'm meeting him face to face, so to speak. So nice to see you, Akhil, and thank you, Dr. Erica. The discussion was great, and I hope all of you enjoyed very useful session. And thank you, Monica, for all your hard work behind organizing the successful NNF meetings. Akhil, do you want to say anything about your global neonatal network, or have you mentioned already?
Thank you so much, Sridhar. So I wanted to, this is a great opportunity. You know, one of the things that we all think about that when I study NEC or Erica studies NEC, we think the natural incidence is 3%. When somebody from Egypt asks us, hey, what is the incidence? What do you do about NEC? And I ask them, what is your natural frequency? And they say 10%. And I go to somebody in Tokyo or Osaka, they say, what is NEC? We only see sepsis. So we desperately need a global organization to look at what are the regional differences. And it is quite possible that by just looking at regional differences in the incidence of a problem, many answers might come up. The second thing is that the incidence of the mortality in neonates is 20%, which is the same as the mortality in the elderly. But babies don't vote. So the incidence, the importance our societies give to neonatal mortality is far lower. So when we talk about, say, I was talking to a person this morning in the Rotaract Club, and the question was that, you know, our donations are small. They're $30,000. And I told them that you're interpreting it in terms of what we could do for the elderly. But $30,000 is a huge amount to make a change for a newborn. Maybe 5 bag and mask ventilations could add 70 years of life or another functioning brain. So that's why we need a global organization. That's what I wanted to say. Please join this organization. There is no fee. All we need is your voice. And we need to think together as to what we can do for the babies.
Thank you. I wish you all the best for that venture. I think you and Dr. Kamlesh are doing a great job, and Dr. Monica and other colleagues are spreading the word here along with me. So appreciate your involvement in this. I think with this, we will conclude the meeting, and I would like to thank Taufik, Lillian, and the KIC team for their great support as well, and Fadi, of course, for all his behind the scenes work, literally behind the scenes as well. So appreciate you being here, and we will have this recording on the NNF YouTube channel. I think hopefully in the next 2-3 days we should have it up. And those of you who are here, please spread the word so others can gain by watching the channel as well. So thank you so much for being here. Good night.
Good night. Thank you, everyone. Good night.
Thank you, Hafiz.
Thank you,
I
everybody.
forgot to thank you. You are our own team, so even though you
That's
are
all right.
invited.
It goes without saying.
That's true. Thank you.
Thanks again. Yeah.
Thank